My name is Kelsey Lloyd. I'm a medical writer in the Regulatory and Scientific Affairs Department. As part of my job, I write clinical study protocols, including I've worked on several for kidney transplant indications. So in talking about clinical trial design, normally we think about clinical outcomes endpoints, which are endpoints that measure how the patient feels, functions, or survives. But there's also a kind of endpoint called surrogate endpoints, which predict but don't measure directly those clinical outcomes endpoints. And these can be useful when it would be unethical to run a clinical outcomes study, when a clinical outcomes study would take a very long time, or when the benefit of the surrogate endpoint is well known. For example, if blood pressure is used as a surrogate endpoint to predict something else. This is relevant to kidney transplant trials because the clinical outcomes endpoints in kidney transplant are patient survival, graft dysfunction, and biopsy-proven rejection. So the drugs we have now for kidney transplant work very well in the short term. The 1-year patient and graft survival rates are both well above 95%. However, there have not been any new drugs approved in kidney transplant in well over 10 years, and long-term immunosuppression with current drugs has well-known negative effects. So this is where surrogate endpoints in kidney transplant come in. So instead of having a 5-year clinical benefit endpoint trial, you can have still a 5-year trial, but with a data cut after 1 year so that the surrogate endpoint can be assessed. And then with that surrogate endpoint, the sponsor can apply for accelerated approval. Then the drug goes onto the market under accelerated approval conditions. Meanwhile, the trial is still ongoing, collecting more follow-up. Once everyone has 5 years of follow-up, the drug can then undergo traditional approval using the clinical benefits endpoints. What are these markers exactly? So originally there was some interest in using single endpoints like eGFR or proteinuria, but graft dysfunction is a complex condition and might not be captured by one biomarker. So there's a number of groups currently looking at composite markers, combining different measures to get a better prediction of long-term outcomes. So one composite measure is the iBox, which is a combination of eGFR, proteinuria, donor-specific antibody, and histology measures. So this was originally created by a French group, but currently the Transplant Therapeutics Consortium is sponsoring it to get it validated as a biomarker by the US FDA. So what that means is that once it is approved, it will be available to anyone in the transplant community to use in their FDA submissions with no additional questions asked about if this is a good endpoint. Earlier this month, CTI submitted data and analysis package to the FDA's biomarker qualification program, and we're hoping to hear back later this year on if it's accepted.